Hey guys, this is Mr. Mahmood and you are watching the third and final lecture for the cells unit. We've gone over the basic idea of cell theory and the difference between a prokaryotic and eukaryotic cell. Now we're going to focus in and take that information kind of to the final level in terms of basic cell structure and what you need to understand about cells to follow what you need to follow over the course of the remainder of the school year. This concept that we're going to talk about really gets a lot into this early civilization, the idea of early Earth's history and how the first organisms may have kind of branched off to form some of the more modern organisms that we have today. Again, this is a theory of an idea of how things have progressed over time in terms of life on Earth may have started, and then that starting of life has progressed to what we see today. Theories, again, are a very strong scientific term, a highly supported scientific concept. But it is free for debate. It is something that is malleable, that can be changed. If new discoveries are made later, theories evolve and change with them. So again, this is not something that you necessarily have to believe to be correct, but it is important that you understand the scientific explanation of how cells and different categories of cells may have evolved. And that's with this concept that we consider endosymbiotic theory. So we're going to talk about the scientific idea of early Earth and the first organisms and how they might have evolved and branched off. Then after we get into a basic understanding of how it's possible, how this connection may be between prokaryotic and eukaryotic cells, we'll focus in on the eukaryotic cells and talk about some of these major organelles that you're going to need to know about very well over the course of the school year. And finally talk about the idea of levels of organization, how an organism is definitely based at the cell level, one of the 8 characteristics of life. All living things are made of cells, but doesn't necessarily have to stop there. The organism could be unicellular, could be multicellular, and if it's multicellular, then we're talking about different levels of organization. So let's go ahead and get started with this idea of the theory of how things might have evolved from the first living organisms. Before we get there, we have to remind ourselves of the 4 things that all cells have. Remember, regardless of prokaryotic or eukaryotic, all cells have 4 things. They have a cell membrane, they have DNA, they have a cytoplasm, and they have ribosomes, right? So those are the basic components basically necessary to survive. The 8 characteristics of life can all be maintained as long as you have these 4 things. And that's why prokaryotic things like bacteria are considered living, because they can function even though they're very, very simple and have very little going on besides the basics, right? So if we're thinking about early Earth, there was the idea of Miller-Urey talking about how the first organic compounds could be made. Then we talked about some of the more recent ideas about how those first organic compounds could be developed. The idea now is that it was devised as RNA, and RNA was actually developed first, and then the RNA branched into DNA, which branched into proteins, and could have conceivably developed the first living thing, which, as far as we know, based on all the scientific evidence that we have, is a basic prokaryotic organism. Now, the early atmosphere didn't have any oxygen, so the organisms that lived there had to live without oxygen. This was known as anaerobic. So if something is anaerobic, that means it's without oxygen. So conversely, if something is aerobic, it's with oxygen. So the very first organisms that were thought to develop on Earth were about 3.5 billion years ago, and we were considered anaerobic prokaryotes, very simple types of bacteria that function without any oxygen. In fact, they got so good at functioning without oxygen that the presence of oxygen in the future killed off a lot of these things. It was basically like a mass extinction for a lot of these anaerobic prokaryotes when oxygen actually started to develop in the atmosphere. But before then, this was theoretically the only type of organism that could have survived. They had to be able to know how to manage and how to get their energy without the use of oxygen. So these anaerobic prokaryotes were thought to be the only things living on Earth for a fairly long period of time. So this started about 3.5 billion years ago. Then, almost 2.5 billion years ago, the first photosynthetic prokaryote is thought to have developed. This prokaryote is something called cyanobacteria. This is something that's actually still around today, and this single organism is single-handedly responsible, if we follow this scientific theory, it's single-handedly responsible for just about all life on Earth. We all have to have this little guy to thank for giving us the ability to live. And it's because of this organism's ability to go through photosynthesis. And the major product of it, which I know most of you think is sugar, the secondary product is actually oxygen. And it's this thing that created the oxygen that caused that mass extinction that may have killed off a lot of the early organisms, but that massive amount of oxygen is what fueled the survival of all of the modern day organisms that you see now. Almost all of them now are completely aerobic and depend entirely on oxygen in order to function and function efficiently. So these cyanobacteria were the very first bacteria to develop the ability of going through photosynthesis. So however they were able to develop that trait, this trait was developed right around 2.5 billion years ago, about 2.6 billion years ago, and these organisms fueled the oxygen that went into the atmosphere. So once oxygen was into the atmosphere, now we started having the ability of certain bacteria to function in a way that's much more common to us and much more similar to us, using that oxygen, breathing that oxygen, and then turning around and being able to use it for its own energy use. So over time, these prokaryotes became very successful. They flourished. All that oxygen being produced gave abundance and gave a lot for a lot more of these organisms to be made. More photosynthesis was happening, and that led to the real mass development of a lot of these more substantial organisms in the future. Now, this type of cyanobacteria is very basic in structure, but it has all the things it needs to survive. Just like the anaerobic bacteria to start, just like any kind of prokaryotic cell, it has the 4 basic things it needs to survive: DNA, cell membrane, cytoplasm, and ribosomes. And it actually has other things as well. It has a secondary wall around the membrane to give it even more structural support. It has a lot of components within it to give it more strength and composition. It's this structure we're going to talk about a lot as a possible connection with endosymbiotic theory. So these cyanobacteria are prokaryotic, but they're also photosynthetic. So they're able to go through photosynthesis, and they're the producers of a lot of food chains that started to develop in this early atmosphere. Alright, so after a while, and the development of all this oxygen in the atmosphere, some of the bacteria developed the ability to use oxygen and to use it efficiently. So they became what's known as aerobic prokaryotes. So if you have oxygen available and you use oxygen efficiently, you're considered aerobic. So aerobic prokaryotes are able to use that oxygen that was made by these photosynthetic cyanobacteria and start to develop components that give them a lot of success in this environment. And this is a good example of a typical aerobic prokaryote that was developed in around this time. This was close to about 2.3 billion years ago. So around this time we have these different types of bacteria. They both have become very good at what they do. The cyanobacteria are very good at going through photosynthesis, not so good at turning around and using that the sugars that they make and the oxygen for their energy source. But these guys, the aerobic prokaryotes, they can't make the oxygen, they can't make the sugars, but they do really good. They're very, very efficient at using sugars and oxygen to turn around and make its energy. So these two guys separately aren't too great. But if they were to combine, imagine how efficient that organism could be. And this is sort of the idea of what we consider endosymbiotic theory. The idea is over time, these different types of bacteria depended so much on each other and worked together so much that over time it's possible that they could have actually fused to be one within the other, or both fused within other cells to become sort of a giant cell or a supercell, or what we consider a eukaryotic cell. So let's walk through the process. Here's the basic idea behind endosymbiotic theory. We start with a basic prokaryote there at the top. This basic prokaryote again has just genetic information, ribosomes, cell membrane, and cytoplasm. There's no nucleus at this point because there's no need for a nucleus. Remember how we talked about a nucleus as like a helmet? There's no need for a helmet if there's no physical activity going on. It's the activity inside of that cell is very weak, very basic, because there's not a whole lot happening. Now, as these cells progressed and they became a little bigger, a little more advanced, they were able to function a little better, more efficiently, and things started becoming more active inside of the cell. Little by little, we get to the point where we've developed theoretically our first eukaryote. This is the first cell that actually required enough of a protection around its DNA to form a nucleus. And now that we have a nucleus, we are past a prokaryotic cell. Remember, that was one of the key things that distinguishes between prokaryotic and eukaryotic cells. So now that we have our first formed nucleus, we are now considering into our first eukaryotic cell. However, besides the nucleus, it's pretty much just like the others. It's just big and a little more advanced, but it doesn't have any sort of structural components inside that really help it be successful. And this is where we get into the idea of taking advantage of those aerobic bacteria that we talked about before and those cyanobacteria that went through photosynthesis. Theoretically, if enough of these aerobic bacteria were working around these other large eukaryotes, to the point where over many, many generations, the aerobic bacteria actually fused into the eukaryotic cell. Now we have an actual cell within a cell. And that cell within a cell, theoretically, is going to become an organelle that is in all eukaryotic cells today. And that little orange organelle that's referenced there that we're going to talk about is called the mitochondria. So mitochondria, as you'll learn at the end of the year, is an organelle, a part of the cell, that's responsible for taking your sugars and your oxygen and turning around and making ATP with it. ATP stands for adenosine triphosphate. It's your energy source. It's the way that your cells are able to go through chemical reactions. And most of it's being made right here in the mitochondria of a cell. So theoretically, the eukaryotes, the early eukaryotes, weren't doing very well in terms of making that ATP, so their energy production was pretty low. However, once they could get that fused mitochondria, or the fused aerobic bacteria, to become a part of itself, now that cell becomes much more efficient. And now that cell is going to branch off and become the ancestor to all of the organisms whose eukaryotic cells have a mitochondria but no chloroplast. So these branched off, and there are thousands of species of organisms today that have structures like this, that have a nucleus, they have organelles, particularly a mitochondria, but they don't have a chloroplast. And in fact, these organisms do extremely well in most ecosystems. We fall into that category. We, as the animal kingdom, we have mitochondria in our cells like this, but we don't have chloroplasts, and we do just fine, right? But we definitely depend on other organisms that have chloroplasts to make the sugars and the oxygen for us. So the first branch off after the first fusion of a bacteria inside of another cell is this idea of the eukaryotic cell with a mitochondria. And then the other idea is, if this continued on, we had more of this endosymbiosis. Let's break the term down. Endo is the opposite of exo. Exo means out, so what do you think endo means? In, exactly. And then symbiotic, symbiosis is organisms working together. Sym literally means together, bio means living. So symbiotic means living together, and then endosymbiotic means living together within each other. So it's the idea, generally, that cells fused within each other to become more significant cell, to become one more significant cell. So cells kind of fuse within each other to become larger components of the cell. So here's the idea. A typical cell fuses in a aerobic bacteria to become the mitochondria of a new cell, and then we continue this fusion process with more cells that all work together for millions and millions of years. We're talking a very long time here, where there's a whole lot of multiple generations of cells working with other cells, so much so that their genetic information starts to kind of mold in together or fuse within itself. And now we're talking about these cyanobacteria, these photosynthetic bacteria, actually fusing into these cells as well. So now it's already a supercell. It has a nucleus, it has a mitochondria now, or the aerobic bacteria that fused in. And now if it can get the genetic information and get everything needed for that cyanobacteria within it as well, now we're talking about a cell that can not only make its own sugar, because it has the cyanobacteria inside, and make its own oxygen, but now it can take all of that energy, the sugar and the oxygen, and turn around and make significant amounts of ATP with it, with the purpose and the use of that aerobic bacteria, which becomes the mitochondria. So now we're talking about a real supercell, and these are the ones that became our ancestors to all of our producers in our the 6 kingdoms of all living things. And the producers are typically going to be your plant kingdom and algae, which are part of protists. But all of your photosynthetic eukaryotes are thought to have evolved right here from this idea of a fusion of a cell that eventually developed a nucleus with aerobic bacteria and cyanobacteria all becoming part of itself. And there's a whole lot of support behind endosymbiotic theory, particularly because of what they think the aerobic bacteria and the cyanobacteria became, which are 2 organelles known as the mitochondria and the chloroplast. And we're going to spend a little time going over each one of those as we go through some of the other major organelles that are found in a typical eukaryotic cell. So we're going to come back to the mitochondria and the chloroplast and support this endosymbiotic theory with it and give kind of the evidence supporting this idea of endosymbiotic theory. But it's important you understand the general idea here is that eukaryotic cells, which are the larger, more complex, much more advanced cells, are thought to have evolved from prokaryotic cells. And the way it happened is with different prokaryotic cells fusing within bigger cells to become sort of like a supercell. So the idea is a prokaryotic cell got so advanced it needed to develop a nucleus, and it started taking in aerobic bacteria and cyanobacteria to possibly become this kind of a supercell, or just aerobic bacteria to become the kind of a supercell that would branch off to develop the animal kingdom and fungi and other things that are able to take the energy and efficiently produce it into ATP, but maybe not be able to make the sugars. Alright, so let's get into the different parts of the cell, the eukaryotic cell, and eventually we'll come back to the chloroplast and mitochondria. Alright, so the basic parts of a cell can be broken down and made into an analogy, and the analogy that we teachers use all the time is like a city. Imagine we're dealing with a big city and we have all these different parts that allow the city to function. The way we're dealing with a cell, and we have all these different parts that allow the cell to function as well. We have to start, if we're talking about a eukaryotic cell, with the nucleus, because the nucleus, if you'll remember, is what protects and holds that genetic information that's inside. And that genetic information, those nucleic acids, are what give you all of your functioning. They know what your cells should be doing. They have the genetic information to determine all the different chemical reactions that should be happening and produce all the different proteins, which turn around and become enzymes to allow those reactions to happen. So the nucleus is extremely important. It's considered sort of the brain of the cell, which is kind of an ironic term as we're going to get to with levels of organization, because the brain's actually an organ, so you can't have an organ of a cell because the cell is much smaller. But the cell within it has this basic component that has all of its genetic information within the nucleus. We reference that, or I make a reference to sort of like City Hall. City Hall, or your municipal building for the city, is sort of like the nucleus of that city, because it knows what your city should be doing. All of your elected leaders and your elected officials work in that city hall, and they work together, and they're the ones that make the decisions about what's going to really be happening for the whole scope of that city. So just like the nucleus has the genetic information that determines what the cell should be doing, City Hall has all the people in there that are going to make the decisions that will affect the entire city as a whole. And I found this picture. This is actually City Hall in London, and I think it's just awesome architecture. Plus there's a direct connection there if you can think about a nucleus and encompassing, surrounding it. I mean, it looks just like it, right? But this is a perfect example. You're talking about a city so substantial like London, you need a whole lot of people in there working efficiently to really make sure that city is run well. So this City Hall is very big and has a lot of room inside. So very good reference there. So City Hall of a city is just like the nucleus of a cell. It has the information that eventually will control what happens within inside the cell. And remember, this is just in eukaryotic cells. Prokaryotic cells don't need a nucleus. Their genetic information is just kind of free floating. Alright, we also have a cell membrane. This is something that's actually in all cells. It doesn't matter whether you're prokaryotic or eukaryotic. In fact, the next unit is going to be committed entirely to the cell membrane and how it works in terms of maintaining homeostasis. But we'll just introduce it here. So your cell membrane is that border that allows for one of the very important characteristics of life. Remember, the ability to maintain a stable internal environment or maintain homeostasis. It's a cell membrane that allows that to happen, and we're going to explain why in the next unit, but it basically has to do with the size of the gaps in the membrane that allow certain things to come in and out. It's very much like, if we're talking about a city, let's say it's a city that's close to an ocean. It's very much like that city's levee. This is an example of a levee, sort of like a barge or just some sort of a man-made wall that tries to block water from entering, especially if the city is below sea level. It would just be in its own lake, or its own, part of the ocean, if it didn't have something to block the water. So that levee is there to actually block the water from entering in. And if you remember a long time ago, I think 2005, when Hurricane Katrina came in New Orleans, that levee actually broke, and that was a major reason for the flooding that happened there. So the cell membrane, just like this levee, is extremely important to making sure what's going on inside and the inside of that city is maintained, just like the inside of the cell needs to be maintained. Only certain things should be allowed in and out, and it should all be at certain levels to keep the inside the way it's supposed to be. So just like a levee for the border of a city, the cell membrane is very important for regulating certain things in and out and keeping things out that would very much compromise what's going on inside. So that's a cell membrane, just like the levee or the border on the outside. Your ribosomes are responsible for actually making your proteins, and I hope you remember how important proteins are for your cells in terms of specifically being enzymes that allow chemical reactions to happen faster. So the ribosome is the site where these proteins are actually assembled, and we're going to spend some time on this in a few units, but just as an introduction, all of the parts that are going to make the protein, all of those amino acids, are going to be put together in a very specific order based on the DNA, and it's put together right here on the ribosome. So I think of the ribosome sort of like an assembly plant. So like imagine your city had an assembly plant for cars. The cars that are going to get put together are the driving force, right? You like my analogy there? The driving force for the city, because the cars are what drive everybody to get where they need to go. Just like the ribosomes are the assembly plant that make the proteins, and those proteins as enzymes are going to drive all of the chemical reactions in your cells. So both of these, the ribosome and the assembly plant, make the structure that's going to drive the city or drive the cell. So ribosomes make the proteins, assemble the amino acids in the right way so that those proteins can turn around and be enzymes to allow certain chemical reactions to happen more quickly by lowering the activation energy. Just like an assembly plant for a car makes the cars and then allows everybody to drive themselves where they need to go so the city can function properly. So the ribosomes make the proteins to turn around and drive the cell, just like an assembly plant would make the cars that drive the people around the city. The chloroplast and the mitochondria are our last point. So let's come back to this. Notice how the chloroplast picture is actually the exact same as the cyanobacteria. I did that for a reason, because over time, the idea, if you follow endosymbiotic theory, a chloroplast is nothing but a more evolved cyanobacteria. And the way that endosymbiotic theory is supported is by looking at these two organelles in particular, the chloroplast and the mitochondria. Most organelles and most parts of a cell can't really function on their own. They all play a very important role. They're very good at certain things, and they help in the overall functioning of the cell. But if you pulled any of those parts out, most of those parts out of a cell, and left them alone, they wouldn't be able to do anything. And the major reason for that is they don't actually have their own DNA. They depend on the DNA of the cell in the nucleus to turn around and instruct it what to do. It doesn't know what to do, it just has abilities to do certain things, and it's waiting for the genetic information to turn around and make the proteins and enzymes that will allow it to function the way it needs. Most organelles also don't have ribosomes, so they can't turn around and make the proteins that they need anyway, even if they did have DNA. They also don't usually have any sort of an outside barrier to protect itself and allow this kind of homeostasis that we talked about. So most of the organelles that are in a eukaryotic cell can't function on their own. But there are two exceptions to that rule: the chloroplast and the mitochondria. Both the chloroplast and the mitochondria have four things that make them very, very similar to old bacteria that could function completely on their own. They both have their own DNA. They both have their own ribosomes. They both have their own cell membrane and external barrier in order to protect itself. They have actually a double layered membrane. They each have it, so it's not only allowing homeostasis but actually has a significant amount of structural support just on its own. And they both divide the exact same way bacteria divides, through a process called binary fission. So most cells, when we talk about a cell cycle, which we'll get to later, they divide because the DNA of the cell is instructing it to do so, and it divides in a certain way. Not mitochondria and chloroplasts. They go through their own division process, completely separate from the DNA of the cell. They divide on their own by a very similar process to what bacteria go through. We'll talk about the actual process later. But the fact that they do that makes it, again, a very close comparison to these old bacteria. So chloroplasts, because of all these things, could technically live on their own. They could live free and independently, but they're not going to be able to make that efficient of ATP because, remember, they depend now on the mitochondria to do that. But these guys are really good at making sugar, making oxygen, and protecting themselves and functioning completely independently. So the chloroplast is very much like cyanobacteria and is a big, big source of evidence supporting endosymbiotic theory. So now let's get back into this analogy that we talk about for the city. If the chloroplast is responsible for making the sugar, like making the food, I can relate that to sort of like the farm of the city, or because the farm is where the actual food is going to get made or harvested. Animals will grow there, and the plants will be planted there and harvested there. So all of the food for the city could be coming from the farms, just like the chloroplast produces all of the foods, the sugars, for the actual cell, in order for those sugars to turn around and be used by the cell to make ATP. So the chloroplast is very important in the process of photosynthesis and making the sugars and oxygen needed for the rest of the cells to function. Not all eukaryotic cells have a chloroplast, like we don't, but it's okay as long as we are getting the sugars and the oxygen from the outside environment into our cells. That's why we, as organisms that don't have chloroplasts, have to get our food from somewhere else. We're known as consumers because we have to get our food in order to use it as our own energy. If there's a cell or an organism that has cells that have a chloroplast, they have the luxury of making their own food, and they're known as producers. They don't need to go out and find food. They make their own through this process of photosynthesis because of this chloroplast, which theoretically could be a direct descendant of those cyanobacteria from a long, long time ago. And then finally we have our mitochondria. Again, just like the chloroplast, they can depend and function completely independent on their own. They have their own DNA, they have their own ribosomes, they have their own external membranes for homeostasis, and they separate in the exact same way that bacteria do. In fact, a lot of times, this mitochondria, because it has its own DNA, is something that's followed and tracked over generations. Because as we're going to learn later, the mother and the father both contribute to making an offspring. The father gives his sperm, but the mother has the egg. So even though they're both contributing equally genetically, it's actually the mother's egg that's going to be the cell for the new generation. And the genetic information of the father is going to come in to that egg. So because the mother's egg is actually going to become the cell, it's the mitochondria that's from the mom that's going to be the mitochondria of the first cell. So technically, even though you're a perfect combination of your mother and your father in terms of genetics, your mother is who contributed her mitochondria for you. No matter who you are, boy, girl, whatever, your mitochondria came from your mom. So a lot of geneticists don't worry so much about the DNA that's a combination, because there's a lot of variety that comes with how that combination actually happened. But they focus on the mitochondrial DNA, because that changes very little, because it's coming from just mother to child over and over again, without any sort of a secondary parent mixing their DNA within it. So the mitochondrial DNA is something that is usually very easy to track and changes very little, and something that researchers really use quite a bit. So I say all this because it's very important that you understand the mitochondria is absolutely something that can function on its own. It has its own DNA and has all these characteristics that directly connect it. It could be a very direct ancestor or a direct descendant of these aerobic bacteria that were functioning a long, long time ago, right after those cyanobacteria were formed, back before the first eukaryotic cells were developed. So again, these are very strong supports of evidence going with this idea of endosymbiotic theory, that chloroplasts and mitochondria in the modern day were actually cyanobacteria and aerobic bacteria a long time ago that fused in to other cells to become this supercell known as the first eukaryote. Right? And now if we get back to the analogy here, we know the mitochondria is what turns around, uses the sugars and the oxygen to make energy. So we relate that to sort of like the power plant of the city. A lot of times this is referred to as the powerhouse of the cell. That's kind of the standard definition of a mitochondria, because it's what actually takes your sugars and your oxygen, turns around and makes the ATP that is responsible for all of your cell functioning. Every chemical reaction requires a certain amount of ATP, and remember, it's those enzymes that make the amount of ATP needed less than normal so that the chemical reaction can happen faster. So all of these things work together in order to allow for a proper functioning of a eukaryotic cell. Whether you're prokaryotic or eukaryotic, you're still considered living, right? So when we talk about living things, they can be in any range of complexity. If it's a living prokaryote, we know that all prokaryotes are unicellular. That means their entire body is just one cell. Even if they're eukaryotic, we talk about how advanced these cells are, you can still be a eukaryotic organism and be unicellular. And those are organisms that usually fall into certain kingdoms, like kingdom Protista, for example, are eukaryotic, but they're unicellular for the most part. So just because a cell is complex and has a whole lot going on doesn't necessarily mean it's a part of any larger organism. So the whole organism may be unicellular. So in that case, the only level of organization we're talking about hitting here is this first level, which is just considered the cell level. So if an organism stops at the cell level of organization, that means its entire body is just made up of one cell, which would make it unicellular, right? And this is how all prokaryotes are, and there are eukaryotes that are unicellular like this. A lot of the kingdom Protista, even some of the kingdom fungi, are considered unicellular organisms. So their level of organization stops at the cell level. But then if you take multiple cells that are all identical, and they combine together towards a common function, that develops into the next level, what we call a tissue. So a tissue is multiple cells that all do the same thing. Then we have an organ. An organ would just be multiple tissues all functioning towards the same goals. So if we have multiple tissues that are all working together to allow a certain function to happen, that's what we consider an organ. So in this case, the tissue picture is actually the tissue of the lining of the stomach, what was considered the rugae of the stomach. Don't worry about that term. And then the actual organ that you see there has all the different parts is actually a stomach itself, connected to the esophagus and the small intestine. So the organ there in the middle is made of the tissues that you see there that are to the left of it. And then you have an organ system. And an organ system is a combination of multiple organs that are all functioning towards the same common goals. In this case, the stomach, which is an organ, is part of the digestive system, which has multiple functioning to allow for certain purposes of digestion. The stomach by itself couldn't do everything, but it plays a major role in all of the components of the digestive system. So multiple cells make a tissue, multiple tissues make an organ, multiple organs make an organ system. And an organism can really be at anywhere in this level. If it's unicellular, it stops at the cell level. If it's multicellular, then it depends on the kingdom of where it'll end up falling. A lot of the kingdom fungi, for example, they stop at the tissue level because of the way their chemical structure is made up and the way the cells combine together, they stop at the tissue level. Plants and animals typically go all the way to the organ system level in complexity. It has a lot to do with how the organism can function as a whole. The higher you are in your level of organization, usually the more advanced and more complex that organism ends up being. So that's your basic introduction to cells. I hope you guys took it all in. It's very important, again, that you know these basics to keep building over the course of the year, and that's why we put these units here at the very beginning, to really make sure you know what you're talking about as we advance on. Alright, thanks a lot, guys. Take care.